The T7 Virus by Thor Warnken. T7 viruses have a naked membrane. T7 viruses are phages, which means they are viruses that infect bacteria. Most phages are easily distinguished from other types of viruses by their unique head and tail structure. T7 viruses are used extensively in modern molecular biology research. Scientists can manipulate the genomes and proteins of these viruses to study gene expression, protein interactions, and more. The T7 virus's genome is linear dsDNA genome of 40,000 BP, and the T7 virus infects bacteria, specifically Escherichia coli. This diagram shows how the T7 virus replicates or makes copies of itself. First, the tail fiber proteins on the virus's surface bind to specific receptors on the surface of a bacterial host cell. The virus makes an opening in the cell membrane through which it injects its dsDNA genome into the cell's cytosol. With the help of the host cell's machinery, the virus's DNA is transcribed into mRNAs, which are translated into proteins by ribosomes in the cytosol. Some of these proteins are used to build parts of new viruses. The cell also helps the virus make one long DNA strand containing many repeating codons of the virus's genome. This strand is cleaved or cut into several genomes that are packaged into new viruses. Once the new viruses become fully formed through a series of changes called virus maturation, they leave the cell. The viruses are released through a process called cell lysis, which kills the host cell. A vaccine for the T7 virus is not yet available. One thing that I found interesting is that the T7 virus has played several roles in the development of molecular biology. It was the first of the larger phages to be fully sequenced by L.J. Dunn and F.W. Studier in 1983, and the functions of most of its genes were soon identified. It encodes its own RNA polymerase, which transcribes 10 times as fast as the host polymerase.